Hello, welcome to our poster. My name is Quang Tran and I'm from University of Memphis. Today I'm going to present a poster on assembling reeds, improve taxonomic classification of species at the ISMB conference on a specific topics, microbiome COSI, and our poster is number 55. This is sponsored by my advisors, Dr. Vinh Thuy Phan. Is the background in metagenomic studies, species classification and profiling are important problems. On the figures, we see that there are processing steps to go from a complex metagenomic samples to species classification or profiling of sample contents. And with the sequencing data, it's changing the microbiology studies from determining etiology of an infection from patient blood samples to survey bacteria in an environment samples. And here we focus on classification um, approach where they employ shock reads to classify, bin, and profile microbiome genomes that are present in samples. Many of these methods adopt techniques that aim to identify unique genomic regions of genomes so that differentiates them. Because of this, shock reads length may be suboptimal. However, longer read length may improve the performance of classification. Um, in the meantime, longer read produced by current technology tend to have a higher rate of sequencing errors compared to short reads. So it's not quite clear if the trade-off between longer read length versus higher sequencing errors will increase or decrease classification and profiling performance. So here's our approach. Um, in these studies, we mainly focus on metagenomic classifiers. So most of the metagenomic classifiers, including those that we study in these papers, consist of main two steps. In the processing step, a classifier utilizes reference genomic sequence of existing species to build an index or reference tables. The index or reference tables is built only once for metagenomic environment. In the classification step, the classifiers use the index or reference tables to classify metagenomic data in the form of reads and make predictions. So our approach interrupts this workflow by modifying the classification step. Before feeding short reads as input to classifiers, we assemble them into longer reads. So in the figure, it's showing the process of comparing the classifier performance on short reads and longer reads. On the figure A is the standard or traditional workflow of a classifier in which the classifier takes as input a short read dataset and output species that it predicts to be present in the samples. In the figure B show a workflow in which the short reads are first assembled before feeding to classifiers. We tested 7 classifiers and 3 assemblers in these studies. Classifiers were evaluated with synthetic and real samples. Although some tools can work on the strain levels, we evaluate classification results at species levels since most methods still do not provide strain level identifications. Classification performance of synthetic data was measured in terms of precision recall and F1. And here the main data set consists of three main data sets: 10 species, 10S, 100 species, 100S, and 400 species, 400S. Using a sample reads, 4 out of 7 classifiers increase their precision by at most double while maintaining similar recall. You can see at the table. These four classifiers are Kaiju, Clock, Recon, and Metacache. The improvement in performance was most critical for smaller datasets. Here's the overall results of F1 score across 7 classifiers as well as on short reads NA and longer reads get assembled by megahit MH, by metaspace MS, and Ray. So on the figures, metaphylan 2 performance got worse with assembled reads compared to its performance on short reads. As seen in the figures, the overall F1 score is highest when reads are not assembled with metaphylan 2. We also observed that Deutsch and Gotcha did not benefit from assembled reads. Figure also show that the F1 score of Deutsch and Gotcha remained unchanged or decreased slightly with assembled reads. We also observed that F1 score was the highest when reads are assembled by megahit and metaspace. And the link below of the figures where you can find our preprint of the papers. By the result above, I conclude the papers with two implications. First, classification species in metagenomic environment can be achieved with higher overall performance, simply assembling short reads into a longer reads or context. And again, since longer reads, assuming that classifiers may not make a random guess on the classifications, so it may be a good idea to consider utilizing long reads technology in species classification for metagenomic applications. Finally, I thank you for your attention.